So thank you a lot, Athena. Thanks all of you for attending, also for Benedetta for organizing this. And I'm very happy to have this opportunity to present my PhD work, which is entitled Selective Processing. Just a second. Yeah. Selective Processing and Associative Transfer Learning in the Sleeping Brain. And I'm really happy to join the Physiology of Cognition Lab with Athena, as she said, working on anesthesia. And we have also a project pending for funding about the interaction between the heart and the brain during sleep and how it concurs to sleep functions like memory consolidation, emotional regulation, and consciousness. So if you're interested in all these topics, don't hesitate to come and we can discuss later on. I also want to share with you that I'm part of the ALIUS research group, which is an international research group dedicated to the study of the diversity of conscious states. So going a bit away from the standard wakeful state and study consciousness in diverse forms. And also I want to briefly say that Charlotte Martial, who is also part of the group, will publish a blog post on Surviving Death, this Netflix documentary that will be that was about near-death experiences. So I encourage you to check it out and check our website also to have more interviews, for example, between Olivia Grosseries and Charlotte Martial, both from the Coma Science group, about the psychedelics treatment for disorders of consciousness. So we are very excited about also getting into this environment with all of you working on these issues. And the last thing I want to thank also Sid Quida, of course, who was with I did this PhD work, and Damien Léger from Centre du Sommeil et de la Vigilance at Hôtel Dieu, where we got interested in the electrophysiology of sleep. And our research question was trying to address why we disconnect from our environment during sleep. Actually, when we sleep, we are not idle. We have a lot of mental activity going on, and this can be also associated with electrophysiological markers. And our hypothesis was that actually our ability to attend and even encode external information during sleep was depending on the ongoing mental activity, and which can be dreaming or, for example, memory processes. And this would just thus depend on this ability to be connected with our environment while being asleep would depend on the presence of these electrophysiological markers related to sleep functions. And that's I will be topicking about. But first, briefly, I will do an overall introduction about what is sleep and what are the methodological challenges for the sleep study. Then I will talk about studies about sleep, selective processing during sleep, and then about associative transfer learning, where we try to teach Japanese to participants while they were asleep. So what is sleep? So the obvious thing whenever we talk about sleep is to characterize it as a behavioral state where we are behaviorally inactive. And it's not that we are not able to move, it's also that we are less sensitive to external information. So for example, it needs a higher intensity of stimulus to trigger a physiological or behavioral reaction. So it's a state of, so to say, sensory disconnection. And here you have the picture of this painting by Magritte, who is called the reckless sleeper. And why the sleeper is reckless is because this disconnection, both at the motor output, but also from the sensory input, leaves us in a very vulnerable position. And it's asked the question why we enter in this state where we can be very vulnerable to predators, and why it has been evolutionarily conserved in almost all animals and is found in every humans. And so actually there is a lot of going on during sleep. And one remarkable fact is that if we sleep a bit less, the night before we tend to sleep longer. So it suggests that sleep fulfills a nomostatically regulated function. And it's related to cognitive activities going on during sleep. But the challenge for the cognitive psychology is how we can probe these cognitive functions, because traditionally, experimental psychology is relying on assessing behavioral responses, which are lacking during sleep. So one way to overpass this problem is to use neuroimaging, and here electrophysiology. So we can record from the scalp, which is called electroencephalography, where we record the most superficial layer of the brain called the cortex. And we can record also around the eyes for the movement of the eyes. So here, electrooculography, and also from the chin, muscular activity called electromyography. And with all these signals, what we observe is that actually during sleep, there is a diversity of physiological events and states. And so the first big division we have is between non-REM sleep and REM sleep. So non-REM sleep is actually here depicted, and it's further divided in light non-REM sleep and deep non-REM sleep. And it's being identified thanks to the occurrence of specific physiological markers like sleep spindles and K complexes for the light non-REM sleep. Actually, K complex are these large deflections which are most prominent of the frontal regions, which are really also large amplitude, but also at the lower frequency. And when they can occur in isolation during light non-REM sleep, and then they can tend to occur in bursts and then also in trains. And this marks the transition to a deep non-REM sleep. And these signals have been overall associated with memory processes like memory consolidation occurring during sleep. So the second big phase is called REM sleep for rapid eye movements. It's also called paradoxical sleep, because we have here an EEG that is really close to what we find during wakefulness, that is desynchronized, but our motor activity is completely shut. So we have here the muscular tonus, which is really low. And it can be also further subdivided into tonic REM and phasic REM, depending on the presence of rapid eye movements. So actually, most of the rapid eye movement sleep is devoid of rapid eye movement. It's called tonic, and it's up to 80% of the REM sleep. And then the rapid eye movement can occur in bursts, about 10 seconds, and they are called here the phasic REM sleep. And rapid eye movements have been associated with dreaming activity, even also dreaming can happen outside the occurrence of these rapid eye movements. But whenever we have those, if we wake up someone who is having a rapid eye movement, we have like 95% chance that he's having a dream, and also these dreams tend to be more intense. So also, what is important about sleep is that these states are not happening randomly. They follow a structure. And the first big division is between the first part of the night and the second part of the night. In the first part of the night, you have a lot of non-REM sleep and also a deep sleep, which is progressively replaced by the occurrence of REM sleep. And you have also a finer grain scale phenomenon, cyclicity, which is the sleep cycles, about 90 minutes. And there is a progression within these cycles going from lighter sleep stage to a deeper sleep stage. So here, to sum up, we saw that probing sleep cognitive functions is challenging in absence of behavior, but we have neuroimaging and more precisely electrophysiology that can be helped to correlate and see that there is different kind of correlate sleep cognitive function with these markers. And we can also take advantage of the timing of this sleep process and the occurrence of the sleep states and sleep electrophysiological events. Because, for example, if we want to study more REM sleep, we can make an experiment towards the end of the night, which is enriched in REM sleep. And we'll see how we did that just after. So the first study is about selective processing. And actually, we used the same protocol across two studies. That one was in the afternoon naps, which is more closely mimicking the early phase of the night, which are enriched in non-REM sleep. The second one was done in morning naps, which is enriched more in REM sleep. So to present a bit the approach we followed, we used the task induction approach, which was theorized by Sittwieder in 2014. And what he did with his colleagues in this article is that he trained the participants to do a task at wake. So they had to push on the left button if it was a word, and the right button if they heard a pseudo word. And the nice thing with that is that you can train it while being awake, and then keep presenting the words as the participant is falling asleep. And the first advantage is that you can first look while he is awake about the neural correlates of the task. Here, whenever you push a button, you have a contralateral activation of the motor cortex. And then you can use these markers while being asleep to see if the participant is still processing the stimuli accordingly. And also the second advantage is that you can train the participant to do it and automatize it. And we know that during sleep, we have a drop in executive function. So if it's automatized, we can just bypass the need to initiate a new task while being asleep. And thus, we can directly probe the sensory processes here. So what they found using this index, which is called the lateralized readiness potential, is that you have this categorization happening during wake, but also during sleep. So we can decide whether something is a word or pseudo words while being asleep. And the nice thing also is that they could look at what happened just before the stimuli are presented. And to do that, they used a metric called the impulsive complexity, which is an index that correlates with, so to say, the conscious level. So here it's very high during the baseline, during wakefulness, and then it drops during light and even more in deeper sleep stage. And then in REM is regained, because we think that REM is associated with this more complex dreaming activity. And the nice thing with that is they could also correlate trial by trial what happened, the baseline impulsive complexity with this marker of categorization, and draw a positive correlation during wakefulness, suggesting that the more conscious, so to say, you are, the more you're able to categorize external stimuli, which is also true for light non-REM sleep. But interestingly, for REM sleep, the reverse pattern was found, suggesting that the more conscious we have, and maybe the more dreaming activity we have, the less we are able to process external stimulus. So we wanted to dig a bit more into this question, and also not just look if we can categorize the stimulus, but whether we can select some information or one over the other. And to do so, we use a classical paradigm called the cocktail party. So the participants were hearing two different speech streams, one in each ear. One was in French, and it will be referred as informative afterwards. And I will play to you an example here in English.
The sleeping brain is not an idle brain, but can actually treat a lot of information.
And the other ear, they were hearing pseudo-text, which is called Jabberwocky, inspiration by Lewis Carroll's poem, where every content word, so the words that bear the meaning, are replaced by pseudo words. I will play to you an example.
The mere king SOS net and equarias, but partially for relative CLIZ.
And so they heard the same simultaneously, and what the participants had to do is to focus exclusively on the French and completely ignore the Jabberwocky. And I will play you here an example. And try to focus only on the French, on the English here, and forget about the Jabberwocky.
The sleeping brain is not an idle brain, but can actually treat a lot of information.
So it's not that easy, but over the trials, people learn how to do this. And we wanted to see whenever after they fall asleep, whether there is an interaction with the dreaming activity and with all kinds of processes happening during sleep. So how can we know that they were following one or the other stream while being asleep in absence of behavior? What we use is the stimulus reconstruction approach. So it's a decoding method. And I will just briefly present the protocol and explain the relevance of each step in this approach. So first, before, at the beginning of the experiment, they were hearing the same text in both ears. First the informative and then the Jabberwocky. And then they went to the test phase where they were hearing the both simultaneously and had to focus on the informative exclusively. And then they were falling asleep and there were always like new stories coming in. And so the thing is that using the training phase, we could correlate the sound envelopes. So it's basically the variations of the amplitude of the sounds. And we can also extract the filtered EEG signal to build a linear model of how the brain is reacting to the variations of the sound. And using this model, we could apply it to the EEG data during the test phase to reconstruct a predicted sound envelope and then compare it with the actual inputs they were hearing in the left ear or the right ear. And so we drew correlation scores. Actually, we checked in a control experiment at Wake that if you attend selectively on informative or Jabberwocky trial by trial, higher reconstruction score for the informative or the Jabberwocky. So it's very sensitive to the change of orientation of attention. So now to present to you the results. So first here we invited participants to the lab at 7:30 and we asked them to sleep a bit less, up to 30%. So they come to the lab and finish their night. And it's a bit like the Sunday morning when you go back to sleep and you have all these crazy dreams. We wanted to mimic this effect, but do it in the laboratory settings. So out of 42 participants, we had 18 that had sufficiently amount of sustained REM sleep. And here are the results. What we found is that during wakefulness, there is this increased reconstruction score of the informative over the Jabberwocky, showing that we could attend the informative speech selectively, but also during light non-REM sleep and also during rapid eye movement sleep. And we can compare these results with what has been obtained previously during afternoon naps. And so here we replicated these results that we obtained before for light non-REM sleep. And we hadn't enough in our morning naps sufficiently amount of deep sleep. So, but what's interesting for deep sleep in this study made before is that it was not, there is no preferred reconstruction of the informative speech over the Jabberwocky one. And if something is more a tendency to the reverse. But we still wanted to know if something similar to that happens during REM sleep. And to do that, we looked at this subdivision of REM sleep between tonic and phasic REM. So tonic when there's no rapid eye movement and phasic when there is this presence of rapid eye movement. So we took 20 seconds window and what we found and scored them either as tonic or phasic if there were a burst of rapid eye movements. And we have this pattern where the selective amplification of the informative speech is restricted to the tonic REM. And it's very strikingly similar to the pattern of results we have when we compare light non-REM sleep and deep non-REM sleep. So we have here a parallel between what happens during REM sleep and during non-REM sleep. But we wanted to look a bit more what happens with these eye movements. So we correlated the amount of eye movements within one trial, and one trial is about 1 minute. So they were hearing the stories during 1 minute, then they had 4 to 6 seconds break, and then hearing another story for 1 minute, and this in a row. So whenever we looked at the number of eye movements within one trial and the reconstruction score during REM sleep, we have a selective disruption of the reconstruction of the informative speech, which was not found for Jabberwocky. So the more eye movements you have, the less you are able to reconstruct, to process the informative stories. So to have a closer look on that, we can use a sliding window approach. So we can decode the reconstruction score on 4 seconds window that are slit just before the onset of burst of rapid eye movements, during, and then after the offset of them. And what we find is before the onset of burst of rapid eye movements, we have this selective amplification, which is lost, and then we have a regain of selective amplification of informative speech. But here it's a bit messy because with the burst, we have all kind of electrophysiological activity that can be contaminating or measures here, especially during the burst. So what we did is to focus only on isolated eye movements. And when we do this, we obtain a clear suppression of the informative speech, which is very selective because the Jabberwocky speech is unaffected. And we can here compare what we obtain generally during REM sleep as what we obtain at the very onset of isolated eye movements. And interestingly, if we compare these results with what was obtained previously with slow waves, we have also a striking similarity with the selective suppression of the informative stories at the onset of slow waves. So here, despite there are really drastically different electrophysiological events, you can see here on the EEG and the EOG, we have a striking similarity on the kind of selective suppression of external stimuli that is going on. So now I will take another look at this on the data, which is about the dynamics of selective processing. So what happened in one trial, which is lasting about 60 seconds. And here in wakefulness, we see that we have a sustained selective processing over the entire duration of the trial, which is found across both studies. But in light non-REM sleep, it's only transient. So it's only in the beginning of the trial that we have this selective amplification, which is lost afterwards. And we found this across both of our studies. What we obtain in REM sleep is very different. We have actually the reverse pattern. We have no selective amplification in the first half of the trial, but only at the end, we have this delayed selective amplification of the informative stimulus. You can compare that to the deep sleep. So deep non-REM sleep, we have actually what we obtain more closely, which is more closely related to what we obtain for light non-REM sleep with a selective amplification in the first half, and then a tendency of the reversal in the second half that we can be found here a bit for REM sleep. But to be more precise on that, we will further divide REM sleep in tonic and phasic REM as we did before. And we are scored either whether they were belonging to the first half of the trial or the second half of the trial. What we found is that we have this selective amplification overall during tonic REM, which is a bit bigger in the second half. But in phasic REM, interestingly, we have this selective suppression in the first half, then which is reversed in the second half. Interestingly enough, it's not related to the amount of rapid eye movement because the amount of rapid eye movement is the same in both the first and the second half. So it's very independent phenomenon taking part in the selective processing here during sleep. So we can compare this with what we obtained in light non-REM sleep and deep non-REM sleep. And so we have a bit like the mirror image, like selective processing in the first half, which is lost afterwards, and then the reversal, but with the inverted pattern as compared to phasic REM. So now we put a bit more into context regarding the literature. And we know for a long time that we are able to select relevant stimulus from the environment where we are asleep. And here, for example, Formby showed that mothers were more sensitive to the cry of their own child compared to someone else's. It's also true for our own names compared to someone else's name. And some researchers also focused more on the subdivision of REM between phasic and tonic and showed that you have a higher deflection in response to auditory deviance when you are in tonic compared to phasic REM. But they didn't really show in the literature before that you can selectively suppress the relevant stimulus and leave other type of information unaffected. And here our approach was a bit different compared to what was previously done, because we had this ability to present two stimulus stream at the same time and really look at the selection in the same time of one information over the other, and show this flexibility depending on the sleep substage that we are in. Also, what was interesting to observe in our experiments is that you have this striking parallel between light and deep non-REM sleep on one hand, and phasic and tonic REM sleep on the other hand. So actually, Hermes and colleagues looked at the arousal thresholds, so the intensity of stimulus which is necessary to trigger a physiological behavioral arousal, and showed that you have this similarity between light non-REM and tonic REM on one hand, and deep non-REM and phasic REM on the other hand. But here we could, so we had the same kind of similarities, sorry, but also at the microphysiological level, because when we looked really at the moment of eye movements and at the moment of slow waves, we had this selective suppression of the informative stimulus. And what we think it informs us about is the type of mechanisms that goes on for the sensory gating, because what happens during REM sleep is very different than what happens during non-REM sleep. In non-REM sleep, you have, with these slow waves, the periods of cortical silencing that is related to the occurrence of slow waves. So it could gate here at the cortical level the transmission of information, while in REM sleep you don't have these kind of events during rapid eye movement. So we think it's more can be understood as informational gating, the fact that there is a competition for attentional resources between internal activity that is related to dreaming activity and the ability to process external information. But what was also interesting to find is also differences between non-REM and REM related to neuromodulation and to the dynamics of stimulus processing, because we know that there is difference in terms of neuromodulation, which is here between non-REM and REM, where non-REM is more noradrenaline, mediated by noradrenaline, while acetylcholine is more present in REM sleep. And actually, noradrenaline has been related to the alerting system when we follow the different attentional system theorized by Posner, for example, where alerting could explain why we are able to detect new incoming information and attend to it selectively, but then over time we lose interest to it, while the acetylcholine is more related to the orienting system, so the ability to shift attention and it would explain why in REM we don't detect in first hand the relevant information and tune into it, but then over time we could reallocate our attentional resource towards it. An interesting fact is also that it's not just the levels of this neuromodulation which is changing across states, but you have also fast modulations which are occurring during slow waves with bursts of noradrenaline, and also during rapid eye movement with bursts of acetylcholine. So it would be interesting to dig a bit more into these factors that we haven't really looked, but it's just hypothetical at this point. And so the last thing I wanted to draw your attention about for this study is related to the fact that actually we found slow waves also during REM sleep, both during phasic and tonic with different regional distribution. And it raises the question whether they could play a part in stimulus gating here. Also, dreaming activity is not just limited to rapid eye movements. It has been found that there is other cerebral markers that predict the fact that we are dreaming both in non-REM and REM sleep. It would be interesting to look whether these could also explain this kind of informational gating that we are talking about. And the last thing is also about daydreaming, the fact that dreams also could be happening during the day in the form of mind wandering. It has been hypothesized that actually dreaming during sleep is an intensified form of mind wandering. And there is also markers that have been classically restricted to sleep, like slow waves, but which are found also during wake in a local manner, and they were able to explain patterns of mind wandering. So it could be interesting in this in the wakeful state where you're able to report about your internal processes more directly, what's happening if you detect these markers and you selectively maybe gate external information. So to sum up this study, so we showed that you have flexible selective processing of external information during REM sleep. And we think it's showing evidence for informational gating, which is happening during eye movements of REM sleep. And it also raises the question whether we can generalize these results to other markers or other forms of dreaming activity. So I'll just take a bit of water and we'll continue. All right, so now we'll jump to whether we can learn Japanese while being asleep. So actually the idea that we can learn a foreign language is a kind of a fantasy that goes back. Here it's illustrated in the 50s by Discover, where there is the promise that you can learn anything from self-confidence to college math. So is it a myth or can it be scientifically proven? And I'll go back to this experiment that I was presenting you earlier about this lexical categorization task. And what they did is also whenever after the sleep, they were doing a memory test. So participants had to say they were hearing words, either that they heard during a wakeful state, either during sleep, or either entirely new words. And they had to say whether they heard this word before or not. And what they found is that actually, if you present words that they were heard during sleep or entering new words, there were no difference. So they say that actually there are no recollection of them. But if you ask them confidence, they will be a bit more confident in saying that when they were saying that they heard this word before, if they were presented during sleep compared to whether it was never presented. Showing that they have a kind of implicit form of memory, so like an intuition that actually they maybe heard this word before, even if it was during sleep. So we wanted to look whether we can have this kind of learning, but not just for words, but also for associations. And here the association we wanted to do is between one word or also like a concept, like a word, here names, and its meaning. So what we chose is to look at Japanese, and here we designed an associative transfer learning paradigm. I will present it to you briefly now. So first there is a familiarization phase where they were presented pictures with the sounds which were associated to these pictures. So here just the sound of a barking dog. And they were presented this association 4 times, just that they have it in mind. Then they were going to sleep, and whenever they were asleep, we played the sound of the dog with the Japanese translation on top. And then afterwards, we did that during sleep. They were waking up, and they were presenting only the Japanese words with 2 pictures, and they had to say which one they thought was the correct answer, and so give their confidence. I wanted to see if they learned this association between the dog and the Japanese translation for dog. And we had 48 Japanese words divided in 3 lists. So 16 items would be presented only during non-REM sleep, 16 during REM sleep, and 16 were not presented during the sleep, but just at the test phase, just to control that they were not knowing Japanese beforehand. And of course, we also recruited people that were naive to Japanese. So here it's an example of an experiment at the sleep stages they were into. So what we did is we woke up the people in the middle of the night. The first part of the night was a task completely unrelated. And they did the familiarization. Then they go back to sleep. They heard these words with the sounds with the translation on top. And then they woke up and took the test. And what we can observe here is that we obtain more non-REM sleep as compared to REM sleep. Actually, we could repeat this association about 3 times more in non-REM compared to REM sleep, about 40 times in non-REM sleep, and about 13 times in REM sleep. So here are the results. So we found that whenever they were trying to guess which was the correct answer while being awake, they were at 60% chance above, and thus significantly above chance level for non-REM sleep. And they were on chance level for REM sleep and also for control. Whenever we looked at the confidence and compared for the non-REM sleep items, we compared whenever they were giving a correct answer compared to whenever they were making a mistake. Actually, they had no clue, suggesting that even if they learned, they don't have an explicit knowledge about the fact that they learned the meaning of Japanese words. To further stress these points, we did the same experiment while they were with different participants while they were awake. And so we did the same participant, just the moment where we presented the sounds with the Japanese words were done during the wakeful state, and where the words were repeated 4 times. And just with 4 repetitions, they were really higher than what we presented before for non-REM sleep. And when we looked at confidence, they were also more confident compared to items that was never shown before. And they were also had higher confidence whenever they were correct compared to error. So here we have really a qualitative difference between what's happening during the wakeful state and sleep. And here it's more related to an explicit form of memory. And it's also buttress the idea that what we observed before was an implicit learning. So then we looked at what happens when we have the presentation of the words. And what we did is that we logged the cerebral activity in response to the stimulus during sleep. And the interesting thing, we looked at the frontal activity, which is where we have the strongest response and which follows a slow wave rhythm. And we compared the responses for correct items between items that that were leading to a mistake. And we found a difference even after the stimulus was ended, which is more in the third trough here of the slow wave, showing that you have several predictors of the learning while being asleep. So we can focus on what's on the differences here in the gray zones, which are the trough of the slow wave response, and see that we have selective difference only in the third trough. And when we look at the topography, we have difference modulation, which is central, which is localized on the frontal electrodes, so which is really typical of slow waves. And here, if you look at the difference, we have it on the frontal, but also a more posterior, which is left lateralized, which could be related to maybe language processing. And the last thing we could do, which was to look at what happens early in the training, so the first 20 repetitions during non-REM sleep, and more later in the training, in the last 20 repetition, and see that actually the difference between correct and error was more located in the initial encoding of the information in the early training, and more after in the late training, more in the late responses. So the same here, we can focus on on the gray zones here, and show that you have a significant difference in the first half, which is localized in the early responses, and then it's moving towards the later responses later during the learning. How we can interpret that? We think it could be related to the fact that in the initial moment of the learning is more the encoding of the sensory information, which is critical for learning, and then more in the later phase of the learning is the building of an associative networks between different brain regions, which will resonate and oscillate in response to this stimulus presentation. All right, so now we compare to what was obtained in the literature. And actually, in 2019, just while we were making this study, there was another study was being published where they showed that you could do these association learning of the meaning of the words during sleep also. Here between words and pseudo words, if you present pseudo word and pseudo words association during sleep, and you ask afterwards for pseudo words whether about their size, whether they fit in a shoebox or not, actually people were a bit above chance level, showing that you could transfer this property about the size from the word to the pseudo word. And what was interesting is that they also looked at the response over the frontal region in response to the word presentation during sleep, and found this modulation, which is really similar to us, and showed that what predicted the memory performance is the late response very closely as we showed. And here it's more related to slow wave peaks, because they looked at it in deep sleep where you have more sustained slow wave activity, while for us, it was more the slow wave trough, because where our sleep was enriched in light non-REM sleep, because it was in later sleep period. So it was interesting to see that this kind of associative transfer could be evidence in deep non-REM sleep, but also in light non-REM sleep. So what was interesting for us compared to these results is that we show that it could be also generalized cross-modeling, so using pictures and audio. And we also had a more rigorous test about the implicit nature of this memory. It also gives us an insight about how we can understand memory systems. So classically, involving dissociated declarative or explicit or and non-declarative or implicit memory. And declarative memory is more related to conscious processing, and semantic memory was classified as a form of declarative memory. But Henke challenged his view and said in 2010 that we should understand more the memory in terms of memory processes, where you can form semantic memory in an implicit manner, but it needs a lot of repetition, and it's related to a slow encoding of rigid associations. And we could really look at these dynamics of the process and saw how it's building over time. And also that we can compare this to a more rapid encoding of flexible association. And we observe that also in our data, that when we do the same task during wakeful state, we can have it more efficient learning and more quickly with an explicit form of memory. So also, what's the interesting thing is to think about REM sleep. So what happens during REM sleep, even if we regain consciousness during REM, whenever we present, we can look at the results that have been obtained by Artier and colleagues in 2010, showing that humans can learn associations, but more it was really a conditioning paradigm where they paired a tone either with rewarding odors or aversive odors, and looked at the sniffing response, and saw that whenever you present tone that were previously associated with positive odors during sleep, you have a higher sniffing response as compared to tone that were linked to aversive odors. But they were seeing that in non-REM, but also REM sleep. But the problem is that whenever they tested this in the morning, this response for the non-REM was still there, but not REM sleep, suggesting that we cannot transfer the memories that we form during REM sleep into the wakeful state, which makes sense, so to say, when we think about dreaming, because when we dream, we forget about our dream whenever we wake up most of the time. And what was interesting in that data set is they also looked at the dynamics of learning. So in the early training phase and the late training phase, showing that there is changes in the type of responses that they observed. But they didn't look at what happened during REM. It would be interesting also to see if the learning that happens during REM could be also lost within over several repetitions. Because in a dream, for example, we can remember some memories of what was happening during the dream, but maybe after a few minutes, it can be lost. So just to sum up, so we showed that there is associated transfer learning during non-REM sleep. Also showed that we have evidence for cross-modal implicit learning in the semantic memory. And also it raises the question of what happens for memory formation whenever we are in the REM sleep. So I'm really thankful for your attention. Sorry to for having been a bit long. And also I'm really looking forward to your question and discuss the perspectives of these results.